time for episode 22, Taylor's version. What did we do? We did standing calf raise, then we did the seated hamstring curl, and then we did the leg press calf raise, the leg press, Smith machine squat, Smith machine sissy squat, stiff-legged RDLs. Same as usual. Leg training is generally pretty on point. So let's talk about that. Let's talk a little bit more about hormones. Let's back up the hormones for a little bit. So, kind of hormones. Let's tie it in with the heart. So, our SA node sends a depolarization signal down our atria. It propagates through cardiac muscle through the intercalated discs. How does it do that? Well, it has inherent automaticity and receives input from our vagus nerve and our accelerator nerve, which affect the rate of depolarization. How? Vagal tone hyperpolarizes by sending an IPSP, increasing the delay between EPSPs that are sent by the SA node. So we're hyperpolarizing, takes a longer time to reach threshold and polarize, hyperpolarizing. So the signal of the SA node propagates down the atria, stops the layer of CT, running transversely through the heart. The AV node picks up the EPSP and transmits it down the bone branches through the Purkinje fibers, which go back up in the apex. How? Neurosignals are propagated by sodium influx. So let's tie this all the way back, really. Sodium influx, so our depolarization, and then K plus efflux, which is so basically when we send a neural signal, this is kind of review. We're going to have sodium come in, and we're depolarizing the cell, and the potassium is going to flow out. And when the potassium flows out, both concentration gradients have kind of been equalized, so they have to use the sodium-potassium exchanger in order to reset back to baseline during the refractory period. That uses ATP, and as we know, ATP hydrolysis creates hydrogen. Hydrogen is going to fatigue us because it's going to lower our pH. Could this be, I need to actually ask Professor about this, could this be a source of, this is what they call CNS fatigue? You know what I mean? Your nervous system has to recover from the hydrogen it's producing, the nerve cells. Because we haven't really talked about nerve pH. So if the pH is being lowered in our nerve cells, denaturing whatever proteins are there, that's probably CNS fatigue, and that's probably, they talk about the only reason you really need rest days is to recover from CNS fatigue. Not necessarily peripheral fatigue, because you can kind of train through that sometimes. Though it's probably not optimal. The other source of hydrogen. I didn't even think about that. Let's see. This episode, did I already made a joke about episode 22, Taylor's version? I don't know. It's important. Let's see. So nervous system propagated, sodium influx, yeah. How, let's see, loss of water during exercise. What? Changes in plasma volume affect the blood concentration of a hormone. So how does it do that? The loss of water during exercise through sweat and shifts in fluid compartments is the main way. Well, that's the main way it is. You lose, what is it? Water during, you lose blood plasma during sweat, but the shifts in fluid compartments compensate for that. It's really those shifts that change the hormone concentration locally. And hormones can induce changes in a cell. How? There's a slow method, you can alter the DNA, and there's the quick method, the second messenger system. So with the slow method, basically you have steroid hormone, which can diffuse straight through the cell, bind to a receptor site in the nucleus. We might have to flip back to this. I definitely have to flip back to this, yeah. Binds to SRC. So SRC is the, the steroid receptor complex. It enters the nucleus and then the steroid receptor complex binds to a hormone responsive site on the DNA, activates / suppresses specific mRNA genes, and the mRNA signals or doesn't signal for the creation of a specific protein within the cell. And there's this thing called polymerase II, which opens the DNA and transcribes for RNA. And that's kind of